Hey guys, this is Kendall Terry, and this is active transport for cells. So let's look at this. So when we're looking at active transport in cells, we're talking about things that require energy, and the energy that they require is in the form of ATP, which is useful cellular energy, or the type of energy that cells want to use. And it's movement against the concentration gradient. And we're going to look at some other examples of that right here. You can see the example on the screen going from low concentration to high concentration with energy applied. We call that against its concentration gradient, and that is active transport. There are two main methods of active transport. The first is pumps. So anytime you see a protein that's in a membrane, it's referred to as a pump. You know that it's active transport. The second is cellular, as a change in the cellular membrane, and this is through the process right here on the screen, endocytosis, and it's a process in which a cell will engulf things in its environment. So for active transport in pumps, they require a protein channel, and that pump requires ATP. Of course, it's movement from low concentration to high concentration. You can think of it as going uphill. In class, we talk about riding a bike. When you ride a bike uphill, it takes energy. And that's the same thing you can think of in active transport. You're going uphill, and if you're going uphill, you have to apply energy for that process to take place. And it works in the opposite direction of facilitated diffusion. Facilitated diffusion also uses proteins. But these proteins are more in the line of just channels, which are holes that stuff goes through either direction many times. But in active transport, no longer is it just an open channel. It's some kind of channel that has to open and close, and that opening and closing is causing energy, and it's also got a motion of pulling things against their concentration gradient. And you can see examples of active transport. So energy, by the way of ATP, forces materials through a protein in the membrane against its concentration gradient. Here's an example of calcium being forced across that membrane with the addition of ATP. Very important process in the cell, by the way. There's two types of active transport pumps that we concern ourselves with really in biology. The first is called proton pumps. The second is called sodium-potassium pumps. The sodium-potassium pump is the one that you see most often in examples. The proton pump is the one that we see in photosynthesis and cellular respiration. Both types require energy for them to work. So when you're looking at a proton pump pumps hydrogen, you can see that here on the screen, from one side of the membrane to the other. It's usually going from an intercellular position to an extracellular position, and this may be, instead of extracellular, this may be kind of like a space between two membranes, is what you'll see in cellular respiration and photosynthesis. So that hydrogen is going against its concentration gradient. Primarily inside of the cell is a negative environment. Primarily outside of the cell is a positive environment. So pumping hydrogens out, which is a positive, into a positive environment, that goes against its concentration gradient. So you need ATPs for this to take place. In the sodium-potassium pump, you're pumping 3 sodium out and 2 potassium in, and you can see this in the little video that we've got going here. So there come the 2 potassium in, and there go the 3 sodium out. And you can see that it requires energy for this process to take place. When you have a change in the cell membrane, which is the other type of active transport, this is called endocytosis. It takes in or engulfs large materials. Cellular membrane moves in until it encapsulates materials, becoming a vessel. So the opposite of that is exocytosis, which is getting rid of large material. And both of these processes take energy. So vessels fuse with the cell membrane, releasing contents out of the cell, and this could be waste and digestive enzymes, and we call this exocytosis, or an exiting of the cell. Here's an example of exocytosis. So we've talked about some of this in class. We'll go into more detail with parts of this later. But RNA binds with a ribosome and makes proteins in the rough ER. And we've actually talked about that process. That shouldn't be new to you. That protein that is made at the rough ER will be transported through the rough ER until eventually it'll be ready to leave the cell. It'll go through the Golgi apparatus where it'll be packaged in a vessel, and a vesicle ready to leave the cell. and then it fuses with the plasma membrane and kicks whatever that is out. That may be an enzyme for digestion, whatever. These processes require energy to take place. A protein that binds to a specific signal molecule, enabling the cell to respond to that signal molecule, is called a receptor protein. Now, these proteins are very important to the cell functioning as well. So an example, a muscle of a person exercising could not contract without receptor proteins and signal molecules that tell the muscles when to contract and when to relax. If you look in your book, page 84 and 85, this can cause 3 changes in the cell. This can change in the permeability. We're talking about transport. If you've got a receptor protein that is attaching a signal, this could cause the cell, it could cause gates to open, it could cause active transport to start, but it could change the ability for stuff to come in and out of the cell. The second is just what we call second messenger. So the receptor protein gets a signal, it turns on another protein inside the cell that goes and does something else. That's called a second messenger. It could cause activation of enzymes and ion channels to be open, so on and so forth. And then enzyme activation. This could cause chemical reactions in the cell to speed up. So there's been a signal. It causes enzymes inside the cell to all of a sudden start doing some chemical process of breaking down something or building something up so that the cell can function in its environment. And all this is related to proteins that are embedded in the cell membrane. In this case, they're called receptor proteins.